Hello, everyone. Welcome to this week Neurospritz talk at Sano. And today we have Maria Grazia Puxeddu, which is from the Indiana University and from the group of Ola Sporns, and is going to give a talk about multimodal and multi-subject model organization of human brain networks. So, Maria, I leave you the floor to you.
Okay, thank you very much for your invitation. So the project that I'm going to talk about today is a collaborative effort between me, Rick Betzel from Indiana University, Olaf Sporns from Indiana University, Laura Stolfi, that was my advisor during my PhD in Rome, and Josh Faskowitz, which is a postdoc in Olaf Sporns lab, but is moving soon to NIH. So yeah, as Alex introduced me, I took my PhD at the University of Rome La Sapienza, and my focus there was on studying modular organization in brain networks. And but recently I moved to Indiana University of Bloomington, and I'm working with Olaf Sporns right now. And I'm still trying to characterize brain networks organization. So as you probably know, to characterize brain network organization, there are several approaches that we can use. So the first approach, we can investigate the network at a local scale. So we can characterize the node centrality, for example. We can characterize how much nodes are important within the network. And then there is a more global scale where we can see how, for example, we can consider the network as a whole and studying global principle of the brain network. But what I like most is studying the brain network at the mesoscale level, which is located between the local scale and the global scale. And what I like to do is characterize the modular structure of brain networks. So we know that brain networks are modular. That means they're composed by these groups of nodes that are highly connected between them and scarcely connected with the rest of the network. And why are we interested in studying this mesoscale structure of the brain? For example, we can use this to reduce the dimensionality of the network. So for example, we can study a new network which is composed by supernodes that are the original modules of the network, and study a lower dimensional network. Then we can, for example, using this modular structure to identify sets of nodes that are functionally related. So we can hypothesize that, for example, nodes within the same clusters do the same function in the brain. And then we can use the modular structure also to identify special nodes. So for example, nodes that connect modules, which we call connector hubs, or other special nodes. And also, most importantly, it has been shown that the modular structure of the brain varies with disease, cognitive states, and time. So it's important to characterize this modular structure. How can we? So now the question that we want to try to answer in this paper is, what's the relationship between the modular structure in anatomical network and in functional network? So we know that the brain, when studying the brain networks, we can study two different types of brain. The anatomical networks that represent the physical pathways of the brain. So for example, these two nodes will be connected if there's an anatomical link between them. And then we have functional networks where we see the brain activity at the node level, and we can draw a connection between two nodes if the brain activity is somehow correlated between these two nodes. And these two types of networks have very different topology. So, for example, anatomical networks are sparse and are made of only positive weights, while functional networks are usually full connected matrices, and that can have both positive and negative weight. So what our question was, how can we relate modular structure of anatomical networks to the modular structure of functional network? And to do that, we start from the very beginning. So what's the easiest way and also the most used way to find assortative communities in the brain network is the modularity maximization. So how does it work? We want to find the community structure, the partition of the brain that optimizes this quality function. So Q, in this equation, Q is our quality function that we want to optimize. Aij represents the weights of the matrix. So for example, the weight connected node I to node J in the matrix. Then we have a null model Pij, which represents the weight connected node I to node J under a null model. And then we have this other term, which is a delta function, where Ci represents the module to which node I belong to. So, and this function is equal to 1 if nodes I and J belong to the same clusters and 0 otherwise. So in the end, this function, by optimizing this function, we have the partition that give us the highest increase of this difference. So the partition according to which our matrices beats the null model better. So now the question is, how do we choose the null model? There is no right choice for the null model. We can have different solutions for that. So for example, we can assume we can use an Erdős-Rényi null model where connections are just random. We can have a Girvan-Newman null model where we preserve the degree sequence, or we can have a geometric null model where we preserve special relationships, or the POTS or uniform null model where every pair of nodes have the same weight in the null model. So there is no one correct null model. It really depends on the research question and on the network we are using. So for example, one general guideline is that for structural network that, as we said before, are sparse and positive, the Girvan and Newman null model is pretty useful, while for functional networks, it's better to use the uniform null model because their correlation networks and these functional networks are full and have positive and negative weights. And another thing is that we can add a resolution parameter in this equation that scales the importance of the null model. So the lower it is, the more likely is that the weight connecting node I to J beats the null model. So it's more likely to have more nodes in the same clusters because we can easily beat the null model. While if the gamma parameter increases, it becomes more harder to beat the null model. So what we have, if we run the modularity optimization with different gammas, is that for lower gammas, we obtain these very big clusters, very big models of the brain. And as we increase the gamma parameter, we obtain a finer modular structure of the brain. Again, even in this case, there's no a correct values for a correct value for gamma. So what we usually, like a good approach would be consider different gammas and look how the brain networks organizes at different scales, from the coarser to the finer. Okay, and this, yeah.
So these big and small squares does not mean that these are the only clusters you found.
No, these
just
are
an example.
Yeah, this is just an example.
Okay.
Of what we obtain if we run modularity optimization on a functional network with different gamma values. So okay, but here, so we saw how we can find modules in one network, but we want to find modules in two networks, in the functional networks and structural networks, because we want to study their relationship. So one thing that is useful is knowing that there is a model, a multi-layer model for modularity maximization. So assume we have 4 layers of a network, which could be, for example, 4 subjects or 4 time points, or just different realization of the network. So what we could do is put in the 4 layers, so each one of these network, along the diagonal of a bigger matrix. And ideally, we want to find, for example, a community in layer 1, and we want to see how it increases or decreases in layer 2, or if it disappears in layer 2 or layer 3. So what we want to do is tracking communities across layers. But if we run modularity maximization on this matrix, what we would find is that we have some communities in layer 1 and some communities in layer 2. And they are not related because there's no reason for the nodes in layer 2 to be in the same communities in the nodes in layer 1, because they're not connected anyway, so they wouldn't be the node model. So what we could do is adding a weight between the layers that make the nodes link to themselves across layers, and that helps to find consistent communities across layers. So in this way, we could see how communities shrink or increases their size across layer. And we can put this omega resolution parameter in different ways. So for example, this is the temporal way. So every node is connected to itself across layer in this way. So node I in layer 1 is connected to node I in layer 2, and node I in layer 2 is connected to node I in layer 3, and so on. And then there is another way in which we can add this interlayer coupling, which is the categorical way. So in the categorical way, every node is connected to itself across all the layers. So node I in layer 1 is connected to node I in layer 2, but also is connected at node I in layer 3, and so on. So even in this case, there is no right choice for omega, as there wasn't a right choice for gamma before. But what we can do is tuning omega and see, for example, more consistent communities across layers, or so if we increases the omega value, we would see communities very consistent across layers, while if we decreases the omega values, we would see community structures that are almost independent from a layer to another layer. So now the question is, can we use this multi-layer approach to detect models in multimodal datasets? The answer is yes. So for example, we have our structural network and our functional network, and we can put them on the diagonal of a bigger matrix, and we can link them through the omega parameter, and we can investigate this modular structure, how modules vary from structural to functional networks. But what we wanted to do is, so we ask ourselves, okay, but what if we want to discover communities across modality, so across structural and functional networks and across subjects? So we want to find communities across two directions, not only one direction. So what we have here is, suppose we have 4 subjects, and for each of these subjects we have structural connectivity and functional connectivity. What we can do is put them on this big matrix on the diagonal, and what we did is we didn't had only one resolution parameter omega connecting node to itself across layers, but what we did is adding 2 resolution parameters, so omega and eta, and one of them controls the homogeneity of the communities across subjects, while the other one controls the homogeneity of communities across modality. So specifically with eta we can control the consistency of the communities between structural and functional networks, while with omega we can control the stability of the communities across subjects. And so why we did this? Because we think the communities might vary differently across subjects and across modality, of course. And so the idea is that by tuning both omega and eta, we can highlight, so for example, for low etas, we can highlight unique features of the brain functional and structural connectivity, because the modular structure will be very few homogeneous. While if we increase eta, we would highlight shared features of structural and functional networks, because the community will tend to be more homogeneous across modality. And same for omega, but with subjects. So now we have, for example, if we had 2 subjects, we have this kind of configuration. We have eta connecting structural and functional networks, and omega connecting structural networks across subjects, and functional networks across subjects. So now the question is, what happens to gamma? Which neural model we should use to run modularity optimization? Because we said that structural and functional networks need different special neural models. So we saw before that structural networks, with structural networks, it's better to use the Girvan and Newman neural model, while for functional networks, it's better to use a uniform neural model. So which one do we use here? And the other problem is that while the functional networks have weights that in the range -1, 1, because they just come from a correlation. With structural networks, we have positive weights that can be way higher than 1. So what would happen is that if we put together this higher value of the structural connectivity and with these lower values of functional connectivity, what happens is that these higher values would drag the modularity optimization, so would weigh more in the optimization procedure. So how do we solve these 2 different issues? We converted we converted structural networks into correlation networks by computing the Pearson correlation between rows of the matrix. So we have our anatomical network, and we compute the Pearson correlation between each pair of nodes. And we obtain, in this way, we obtain these structural networks. There is a correlation network. So in this way, we have both structural and functional networks falling in the range -1, 1, and we can use the same null model in the modularity optimization. So, okay, finally, we can test our method, our multi-subject and multi-modal modularity optimization. We did that on the NKI dataset, which is an ongoing project collecting lifespan networks, but we actually focused on a restricted number of subjects, which were the healthy adults, because we didn't want like other factors interfering with what we were studying. So, for example, we don't want the age factor altering what we were studying here. We use a 100 nodes Schaeffer parcellation, and we run the modularity optimization with different values, with different combination of the 3 resolution parameters that we have. So, and specifically for each of these resolution parameters, we took 50 values of them in the range W mean, which is the minimum weight of the matrix, and 1. So we had for each of these parameters, in the end, we have 50 samples, 50 values. That turns out to be so in the end, we optimizes this function with 50 per 50 per 50 parameter, which are 125,000 combination of parameters. And so by running our multimodal and multi-subject community detection, what we found is that what we like the algorithm returns a community structure for every subject. So for example, here subject I, and for every combination of the 3 resolution parameters. So, and specifically, the algorithm return a vector of the length, the number of nodes, where each entry denotes to which cluster node I belongs to. And we have these for functional and structural networks for every subject and for every point in the parameter space. So for every combination of the resolution parameters. So now what we want to do is characterizing these detected communities. And to do that, one easy approach is to compute the entropy between functional communities and structural communities. And we can do that for every subject and for every point in the parameter space. And if we do that for every point in the parameter space, what we have is this graph. So we have each one of these points is a value of entropy between structural and functional networks. And we can see how the entropy, which characterizes the variability between functional and structural communities, how this entropy varies in the parameter space. So we can see, for example, that for higher EDAs, we have lower entropy. So it means that for higher EDAs, we have structural and functional communities that are more coupled. And this makes sense to our model because this is what we wanted. And we can also unpack a little bit these results. So for example, in this first graph, we made the average across omega, and we can see how structural and functional, the entropy between structural and functional communities varies across gamma and eta. And we can see that if we go from very low etas to high etas, what we have is a decrease of the entropy. That means that structural and functional communities are more coupled. And there is also a little effect of gamma. So we said before that with the high gamma values, we have very small communities. So, but the point is, if we have very small communities, so for example, that comprise one node or two nodes, then they are necessarily similar between structural and functional connectivity, because these are the same nodes. So we have a little effect of gamma in the entropy also. And then we can do the average across gamma here, and we can see how the entropy varies across omega and eta. And in this case, we can see that omega doesn't affect very much the entropy between structural and functional communities, while what matters is the eta parameter. And so this is just to see how much communities from structural and functional networks are alike across the parameter space. But then we can choose, for example, a specific combination of resolution parameters, and we can see how, locally, how structural and functional communities vary. So, for example, in this case, we choose gamma equal 0.12 and omega equal 0.06, which is more or less here and here in the parameter space. And we can see how structural and functional community reorganize themselves across the eta parameter. And so what we can see is that as we increase eta, the communities here and here tend to be more alike. So what we can do now is also, so in this picture, like nodes are colored according to communities. So for example, for very low eta, we can see that nodes are colored pretty differently, while if we increase eta, they tend to be colored more similarly across modality. So what we can do is also, we can track the entropy between structural and functional communities within subsystem, and we could see, for example, how in the DMN at this scale, in the default mode network, at the beginning, the nodes tend to belong to different communities, while as we increase eta, they tend to belong to the same communities. And but this doesn't happen, for example, for the ventral attentional network, where nodes tend to belong to different communities in functional and structural networks across all the eta range. So that means that different nodes tend to have different coupling across modality. We can also compute the mean of this entropy, and we can see which are the systems that are more likely to share the community affiliation between modalities, and which are the systems that doesn't. So as we said, for example, the ventral attentional networks as high entropy, which mean the nodes belonging to this area tend to have different community affiliation. for all the eta range, while in other systems like the DMN, we reach one point where the nodes belonging to the system have the same community affiliation between modalities. So another thing, so this is what we have if we do a single scale analysis, but now we were interested in seeing if there exist recurrent patterns of variability across all the parameter space. So what we did is we pursued a principal component analysis approach. So again, we can compute the entropy between structural and functional communities for every point of the parameter space, and we would obtain this entropy matrix, which has dimension number of nodes times number of parameters. And what we can do is we can decompose this network, these metrics, through PCA, and we would obtain two matrices U and V. So that are the principal component scores and principal component coefficients. So what we find in U is that every column has dimension number of nodes, is a vector of dimension number of nodes, and we can project this vector on the cortex, and we can interpret it as a mode of variability. So as a mode of entropy between structural and functional communities. While in V, the columns of V have dimension number of parameters, and they reveal where this mode of variability contained in U happen in the parameter space. So we pursued this analysis and we retained only the first 5 components because they explained most of the variance. And what we found is this thing. So we have every column here is one of the 5 components. And in the first row, we have the principal component coefficients that we said we can project on the parameter space and interpret them as where the mode of variability happens in the parameter space. And then we have in the second row here, we have the principal component scores, which we said we can project on the cortex, and we can interpret them. How likely is that a node here has a different community affiliation between structural and functional network. So for example, if we look at the first component here, we can see that this component is more expressed at high etas, and in this space of the parameter space, we have these scores. So we can see that the ventral attentional networks is highly variable, because it has higher scores, while, for example, the control networks is less variable because it has lower scores. And we can look at the other principal components, and we can see how they occupy different spaces in the parameter space, and they have different modes. They present different modes of variability between structural and functional connectivity. So that means that there exist recurrent patterns of intermodality variability, and these are encoded in the parameter space in a non-random manner. And then finally, we try to validate our model on a second dataset. So we use the HCP, the Human Connectome Project dataset, and we run the same model, and we found, so we run our multi-subject, multi-modality modularity optimization, and we run a PCA analysis, and we found that that the components of the principal components in the two dataset correlates pretty well. So this is just a validation. And even if we take a single scale, so if we choose specific gammas and omega, still we have a nice correlation between the human connectome project entropy and the NKI entropy. And these somehow make our model robust. Okay, thank you for the attention.
Okay, then we have you don't see them, but there are other people.
Ah, okay, okay.
Yeah, we are more.
Okay, nice.
Okay, thank you. Interesting thoughts. But so the take-home message is that we can define like a connectome of connectomes.
So
This
yeah,
hyper, how to say, multi-layer clustering of different modalities and different samples.
yes. Here the take-home message is, first of all, we are providing a new method to study community structure across 2 dimensions, because until now we can only track communities across 1 dimension with the classical multi-layer approach. Here we are providing a means to detect communities across 2 dimensions, which could be subjects and modality, or could be subjects and time, could be, well, in this case we focus on subjects and modality. And then the other take-home message is that the relationship between structural and functional networks is not univocally described from one community assignment, but it varies across the parameter space. And with our method, we could highlight specific features of the modality and also shared features between modalities. And we can do that by tuning the eta parameter here, which tends to highlight specific features if it is low, or shared features if it is high. And then what we could do is also we can apply, so in this case we applied this model to a very restricted sample of subjects, which were healthy adults, but it would be interesting also seeing how these modes of variabilities vary with age or with disease. And so this could be our next steps.
Okay. You have questions here or someone online?
Yeah, as usual, I have lots of questions, but maybe I can okay, let others go first. But if not, yeah, I will.
Yeah, I also don't want to be always the one asking.
Sorry?
I also don't want to be the one always asking questions.
Yeah. Well, anyway, so maybe I start, and if people just comes up with questions, then interrupt me. I had one, but you sort of answered it already, which is how these eta, omega, and gamma parameters could capture diseases, but you already asked this one. You answered this one. So I have a question, is that you said you use the Schaeffer atlas, right? Which, if I'm not, it's more cortical, right? So it captures cortical regions. So I guess you are using the same atlas for the structural connectome. Is that right?
Yes, we are using the same atlas for both structural and functional networks.
And my question is, in fact, is always the same question when I read all these types of papers and listen to all these types of talks, that if you are only using cortical structural connections, you are using a lot of other connections, right? You're using subcortical connections or even deeper connections. So, and right?
No, I'm not using subcortical, only cortical.
Yeah, so you're missing missing a lot of connections of the brain. And you are, not you specifically, but in these types of frameworks, you are trying to draw relations between functional and structural. But if you're not taking into account deeper connections, are we not missing like half of the story? Or it's just we expect it adds up also?
Yeah, this is an important point. So specifically, I know there's a postdoc in my lab now that is focusing on integrating subcortical connectivity into cortical. Like we really want to try to expand our analysis and include also subcortical nodes. So this is an important point. But yeah, in this case, we only focused on cortical.
Okay, that's cool to know.
This is a good question. It is important to like extend our models and integrate also subcortical regions because they play an important role in the brain.
So what you're saying is that it's not only just changing the parcellation scheme, you have to do other things also.
Yeah, I mean we didn't dig into it, but I guess yes.
Okay. Okay. And yeah, the last one is, you said that you're doing this in 2 dimensions, but is this extendable to n dimensions? For example, capturing, I don't know, genetic networks also, relations industry.
Yes. In fact, we can be as creative as we want in placing these coupling parameters. We can find very different configuration of these coupling parameters. We can add other types of networks. The important thing is though finding null models that can be adequate for like multi-networks. So, but yeah, in principle, you can add every kind of networks here and link them as you want with these parameters, and it depends on according to your research question.
Okay. Yeah, very cool. Very nice presentation. Yeah. Cheers.
Maybe I have it's more a brainstorming than a real question. So this entire thing is based as a something like a higher order. I think this MOOCA was inspired by this Louvain modularity.
Yes.
Which is something that we know like on the beginning of 2000. Now, would it make sense to use something more modern like this topological data analysis with multiplex, simplex?
Yes. In fact, this is only one way to characterize the organization of the brain networks. So I would suggest using this approach if we are all interested in finding assortative communities. So if we want to find communities that are highly integrated among them, scarcely connected with the rest of the network. But this is only one way of seeing the organization of the brain networks. There exist different ways, and to study these different ways, this approach is not the best one. So there could be other approaches that would be better, like the topological data analysis and studying higher order interaction. So that's my point of view. Like this method is useful if we are all interested in assortative communities. Otherwise, it's not ideal.
Okay. So good to know from. Now, from my side is all. I don't know here. All the other connectome people.
Okay.
No. Okay. No.
Yeah. Oh, sorry.
You have another one.
Yeah, but this is very few. Then very brief. What exactly is the entropy measuring? Perhaps I missed it, or I don't remember anymore. But like you're computing the entropy between what?
I'm computing the entropy between the functional communities and structural communities. So
okay.
this entropy measure, see, for example, if we take this first node, if this node is assigned to the same community in functional and structural networks, then the entropy is 0 because it doesn't vary. While, for example, if we take this node here and we see it is green on the functional network and it's yellow on the structural network, then the entropy is high, and it's 1. So this entropy measure is low when structural, and so we can compute it for every node in the community affiliation. And if globally the entropy is low, that means that globally functional and structural connectivity have the same community affiliation. While if it's high, it's the opposite.
Okay.
Yeah, thank you.
Thank you for the talk. It was. I mean, we are all doing this kind of thing, so that's why maybe no further questions, because we are working exactly in this very similar things.
Okay, cool. Okay.
And yeah, have a nice weekend.
Yeah, you too.
Even without the Corpus Christi.
Yeah, have a nice holiday. Okay, bye.
Okay,
Bye.
Bye.
Thank you.
Bye. See you.
See you.
Bye.
Bye, Marin.
Bye.